Hello everyone, welcome to episode 9. We have now formally entered the world of sensory and perception. In this episode, we're going to take a look at, well, obviously, vision. Let's take an overview of this episode. First, we're going to learn about sensory and perception in general, and then take a look at vision. Then, we're going to get into the details about the eye and the photoreceptor cells. In the later episodes, we will cover the next steps of vision, sense, and perception. Let's get into sense and perception. What is the difference between sense and perception? Well, on one hand, sense is the physiological process of detecting external stimuli. In this process, we are using our sensory organs. For example, when the retina detects light, it reacts to light. This is a process of sense. On the other hand, perception is the psychological process of interpreting external stimuli. In this process, we are mainly using our brain. We not only need to detect the stimuli, but we also need to have attention, memory, and other cognitive functions to help us. Only then can we make sense of the stimuli. What are they? We can perceive what they really are. For example, when you're looking at this slide and recognizing the words, this is a process of perception. There are various types of stimuli and senses. We can generally divide them into physical senses and chemical senses. Physical senses include senses about mechanics. These are gravity, motion, pressure, sound, etc. We have electromagnetic waves, which is light. We also have electricity and magnetic field, and we have temperature. The other big class is chemical senses. Let's take a look at the senses that we humans can sense. Well, we sense gravity by vestibular reception. The vestibular organs is a small organ inside your ears. Motion, such as shaking your head or moving forward, are also sensed by the vestibular organs. External pressure, such as touch, can be sensed by our skin, and internal pressure, such as the pressure between our joints, can be sensed by the touch receptors inside our bodies. This is a sense called proprioception, which gives us information on the location and position of our own bodies. Proprio just means self. And sounds, of course, we hear it by audition, which is just a fancy word for hearing. Light is, of course, sensed through vision. Temperature can be sensed by thermal reception, which is also a function of our skin. And we have various chemical senses. For example, smell. We smell things by detecting the molecules. Taste. We taste things by detecting the taste molecules. Pain is actually also detected through chemical molecules that are released when we are wounded. And itch. Itch is a combination of various chemical senses, such as sensing histamine and other molecules. These are the major senses of us, human beings. Some other animals can have other senses, which are really magical. For example, in terms of motion, fish have a lateral line system. This is a system that allows them to detect the motion of water. Fish also have baroreception, which allows them to sense how deep they are within the water. In terms of sounds, bats have echolocation, with which they can sense the position of the prey instead of just the sound. Some animals are capable of seeing infrared and ultraviolet light. This is called IR or UV vision. Some animals can even sense polarized light, which means light that does not oscillate in all directions, but rather oscillates in one focused direction. Some animals have electroreception, which allows them to sense the electricity. Some birds have magnetoreception, which allows them to sense the magnetic field of the Earth. This can help guide them in their migration. Some animals, such as snakes, have infrared detection. They can see the infrared lights, and therefore they can see the temperature of the things around them. And some insects, such as mosquitoes, can sense the carbon dioxide emitted by living organisms. This can help them find things to suck blood. These are some of the physical and chemical senses that animals and humans have. There are also some non-conscious senses, which means that we are not actually aware that we are sensing them. For example, we have baroreception and chemoreception in the aorta. There are receptors in the aorta that allow us to sense the blood pressure and the CO2 concentration in our blood. This can help us determine whether we need to change our respiration rate and heart rate. However, we are not aware that we are sensing them. Let's take a look at the 6 major senses in the human body: vision, audition, equilibrium, olfaction, gustation, and somatosensation. These senses share some common pathways, which means each of these senses have a unique information passing pathway. However, these pathways have some things in common. Every pathway starts with the sensory organs. For example, the eye, the cochlea, the vestibular organs, etc. And then nerves begin in the sensory organs and lead into the central nervous system. These are usually the cranial nerves, which are abbreviated CN here, or sometimes the spinal nerves, such as in somatosensation. Then most nerves terminate in brainstem nuclei. Those are located inside our brainstem. The brainstem nuclei in turn transmit information to the thalamus. There are various nuclei in the thalamus, for example, the lateral geniculate nucleus, LGN, the medial geniculate nucleus, MGN, the ventral posterior nucleus, VP, etc. And then the thalamus sends information to the cortex, the primary cortex, which are named V1, A1, S1, etc. And the primary cortex goes to higher level cortices for further processing of the information. From this, we can summarize some common points in the pathways. First, there is transduction via specialized receptors. What is transduction? This is the essence of sense. We need to transduce physical or chemical signals into nervous signals. This process is called transduction, and it is achieved always via specialized receptors, such as photoreceptors in the eye. Next, the pathway has a lot in common. We go from sensory organs to peripheral nerves to brainstem nuclei to thalamic nuclei to the primary sensory cortex, then to association areas or higher level cortical areas. There are some exceptions, and the places where exceptions occur are labeled by an asterisk here. We will explore them in the next slide. There is a preservation of information, especially the layout or relative position of information. This is called topy. Think of topology as you think of the suffix topy. We have retinotopy, tonotopy, etc. This means that a map of the sensory information is preserved. We also have extraction of information. This allows us to go from sense to perception, to truly understanding what the stimulus is. And, of course, every sense can adapt to the environment. For example, if the environment is too bright, then our eyes will adjust. Now let's talk about some of the key differences or exceptions in those pathways. First, the olfactory nerve and optic nerve, which carry the olfaction and optic information, belong to the central nervous system rather than the peripheral nervous system. Also, vision and olfaction do not have corresponding brainstem nuclei. They do not go through the brainstem in order to reach the cortex. And olfaction bypasses the thalamus. It is the only sense that does not pass through the thalamus. This is because olfaction is actually a very primeval sense. Many animals, even the simplest animals, have olfaction because detecting chemical molecules in the environment is an essential sense to survival. Since olfaction was evolved very early in evolution, it does not pass through the thalamus, which is a relatively higher level structure. And at the end of this overview of sense and perception section, we'll take a look at some key questions in learning this. First, transduction. What stimuli are we sensing? How are they converted to nervous signals? What anatomical structures are involved? In terms of information flow, how is the nervous signal passed down in the nervous system? And how is the information preserved? In terms of information extraction, how does sense lead to perception? And from a bigger perspective, how is this sense similar or different from others? How is this sense developed and how was it evolved? Now let's get into vision. In vision, the stimulus that we are sensing is light. Light has several physical properties that we would like to know. Firstly, wavelength or frequency. Different wavelengths or different frequency signifies different colors, well, at least in the visible spectrum. There are other frequencies that we cannot see, for example, ultraviolet or infrared. Inside the visible spectrum, the frequency determines the color. At the end of the highest frequency, we have purple or blue. At the end of the lowest frequency, we have red. This is a spectrum of all the colors that we can see, shown at the bottom of the page. We would also like to know the brightness of the light, which is the energy or the amplitude of the light waves. And we have some other visual informations that are higher level. For example, we would like to know the shape or contour of something that we are seeing. We would like to recognize things, for example, words or faces. Those will help us a lot in our lives. We would also like to sense motion because moving things are very important. Maybe a tiger is pouncing towards you and you must recognize it instantly in order to get away. Now, in all episodes about sense and perception, we are going to see a page like this, a map of the pathway of the sensory and perception system. We begin with photoreceptor cells where light is transduced, and then we go to other types of cells. Then the optic nerves, the optic chiasm, and the optic tracts carry away information. It gives information to non-thalamic brain areas. It also gives information to the thalamus. LGN is a thalamic nucleus. And then LGN gives information to the cortical areas. This is a rough sketch of the visual pathway. We will continue seeing this map throughout these episodes. Question time. Please try to describe the 4-step general pathway of sense and perception, and also delineate some of the exceptions to this system. Okay, here we go. The first step is the specialized sensory organ. In the case of vision, it is the eye. Then, the information is carried by peripheral nerves to brainstem nuclei. Exceptions include olfaction and vision, which are not carried by peripheral nerves and do not have brainstem nuclei. Thirdly, the information is transmitted to the thalamus. Olfaction is the only sense in human that does not pass through the thalamus. And finally, we get to the cortex and different types of cortices. And we get to the cortex and different levels of cortices. Okay, the eye. This is an eyeball. The eye utilizes several properties of light. First, refraction. For example, the cornea and lens are able to refract light. We can see here that the lens is a little convex lens. It is like a magnifier. It is able to focus light onto our retina so that the image is well formed. The cornea is also a glass-like structure that can help us focus light. Next, absorption. Absorption is achieved by photopigments inside the retina at the back of the eye. Light is shown, it is focused onto the back of the eye and absorbed by photopigments. The absorption by photopigments is essential for taking in light for transduction. And we also have reflection, not in our case, but in the case of cats and some other animals. In cats, there is a structure called the tapetum lucidum, which is kind of like a reflective carpet at the back of the cat's eye. When light is shown into the eye, the tapetum lucidum reflects some of the light back. This allows light to pass through the retina twice, and this helps increase the sensitivity of the retina to light, especially in conditions where light is very low. That is to say, in night conditions. Okay, it's time to take a look at some of the outer anatomy of the eye. Let's close up this eye. At the back of the eye, here, we have the optic nerve. The optic nerve carries optic information out of the eye and into the brain. We also have blood vessels, As you can see here, there are veins and arteries going into and going out of the eye. And we have muscles, which are not shown here. We will see them shortly in the next slide. The muscles are useful for moving our eyes around. The eyelid. The eyelid, of course, is something that covers your eye and closes when the eye is not needed. Conjunctiva. Conjunctiva is the white thing that surrounds the eye. It serves as a protective layer. Cornea. Cornea is the glassy stuff at the front of your eye, at the uttermost surface. It serves as a protection as well as a refraction. It has the function of protection as well as refracting light and focusing it. To roll the eye around, we need to have muscles. There are 6 muscles around each eye for moving it around. They are the 4 rectus muscles. Rectus means that they're straight. So the superior rectus muscle straightly pulls the eye upward. The inferior rectus muscle pulls the eye downward. The lateral rectus muscle pulls the eye outward, and the medial rectus muscle pulls the eye inward. There are also 2 oblique muscles. The superior oblique muscle actually pulls the eye inward and downward. The inferior oblique muscle pulls the eye inward and upward. These 6 muscles ensure that our eyes move very smoothly. These muscles are very important in a reflex called the vestibular ocular reflex. We sense the movement of our head using our vestibular organs in the ears. Then it gives information to the muscles around our eyes to help us eyes follow what we are looking at. So if you're looking at something while you are moving around, you can still fixate on it. There are also some muscles that control the movements of the eyelid. For example, the orbicularis oculi is a muscle that surrounds the frame of your eye. When it contracts, it closes the eyelid inwards. In contrast, the levator palpebrae superioris muscle is a levator. That means it is a lifter. It lifts your eyelid upward, opening your eye. Moving on into the inner anatomy of the eye. It's time to take the eye apart again. Okay. So first and foremost, we have the aqueous humor, which is the water that exists behind the cornea, in the front, and before the lens here. So here we have the aqueous humor. It is a watery-like fluid. Next, we have the iris. The iris is the part that has a color. It is actually a ring of tissue that leaves a space in between. This space is called the pupil. The pupil is where the light enters your eye. After that, we have the lens, which we have taken out to show it a moment ago. The lens is a convex lens that can change in shape. It has a vital function in focusing the light accurately on your retina. The lens is made of beta crystalline, which is a protein that is transparent. It is also bouncy, so that it can change in shape easily like a spring. The ciliary muscle is a circle of muscles that surrounds the lens. It is the muscles that help to change the shape of our lens. And vitreous humor. Vitreous humor is the big lot here. It is the thing that fills up most of our eye. Vitreous humor is denser than the aqueous humor because, as you can see, vitreous means glassy, glass-like. Vitreous humor serves to push the retina into its position, preventing it from falling off. It also supports the tissue around it. Okay, now that we have the bare eye, the layer at the back of the eye is called the retina. The retina contains the vital cells for visual sensory. And underneath the retina, we have a layer called the choroid plexus. The choroid plexus contains the arteries and veins that supply and carry metabolic waste away from the retina. Okay, out with the eye. Now a detailed look at the refraction in the eye. We need to bend the light to focus it. This is an illustration of refraction. There are two parts of the eye that help with refraction. One is the cornea. The cornea has a refracting power of around 43 diopters. The other is the lens, which has a refracting power of around 12 diopters, but it can vary since the lens can change in shape. The process of the lens changing in shape to focus the image is called accommodation. When we are viewing something far, we don't need to focus things that much. We don't need to focus it, so the lens should be thinner. When it is thin, it has less focusing power. In contrast, when we are viewing something near, we need to focus the things a lot, so the lens becomes thicker. When we have trouble focusing the light correctly, we have refractive errors. For example, myopia, short-sightedness. In this case, we have too much focusing power, or perhaps the eyeball is too long, so that we focus the image before the retina. The exact opposite is hyperopia, farsightedness. In this case, we have too little focusing power and the image is formed behind the retina. Presbyopia is a case that usually occurs in elderlies. It means that the lens have hardened and have trouble adjusting to either far or short vision. And astigmatism is a case where we are not able to focus the light on the center of our retina. We will talk about it in the next part of this episode. This place is called the fovea. We cannot focus the thing exactly on the fovea. In order to correct these refractive errors, we can use glasses. For example, we can use convex lens for hyperopia. Since hyperopia has too little focusing power, we can use convex lens to help with the focusing. We can also use concave lens for myopia. This is a set of concave lens I'm wearing. So, since I am myopic, I have too much focusing power. The concave lens disperses the light for a little bit so that the focusing power is just okay. Also, we can use bifocals for presbyopia. Bifocals are just glasses that are half convex and half concave. This can help elderlies with both far vision and short vision. And astigmatism can also be treated by using the correct lens to focus the light on the right place. In the bending of the light, we need to be aware that there is a reversal of retinal and visual fields. That is to say, the retinal and visual fields, what we see and what is made on our eyes, are completely different. That is to say, the visual fields, which is what we see, and the retinal fields, which is the image formation on our retina, are completely opposite. They are upside down and left side right. The adaptation in the eye. The eye can adapt to the light brightness via the pupillary light reflex. When there is bright light, the parasympathetic division of the ANS is activated. The optic nerve sends the brightness signal to the pretectal nucleus. It is part of the midbrain. Then, the signal goes bilaterally on both sides to the Edinger-Westphal nucleus. The Edinger-Westphal nucleus sends information via the ocular motor nerve to the sphincter pupillae, which is the muscle around the pupil that helps with its contraction. The pupillary light reflex is a basic reflex. When we shine light, for example, on one eye using a torch, we can see that the pupil of this eye contracts. Interestingly, the other eye also contracts. This is called the consensual reflex. The pupillary light reflex is a very useful reflex in emergency checks. When someone has an injury, we can use this reflex to check whether the brainstem is damaged or not. It is usually intact, or otherwise there must be some serious damage. On the other hand, we can also dilate the pupil. This is achieved through the sympathetic division. There is sympathetic innervation of muscles around the pupil via the cervical ganglion of the sympathetic chain. It activates the dilator pupillae, which dilates the pupil, as its name suggests. Question time. Please select the correct statements. There may be multiple answers. A. When in fear, the pupil dilates because the parasympathetic system is activated. B. Convex lens can be used to treat myopia. C. When we look at something far away, the lengths are relatively thinner. And D. The movements of each eye are controlled by 4 muscles. Please take a moment to pause and think about it. The correct answer is C. A. When in fear, the pupil dilates. That is true because the sympathetic division is activated. The sympathetic system is also known as the fight or flight system. B. Convex lens can be used to treat myopia. That is not true. We use concave lens to treat myopia. C. When we look at something far away, the lenses are relatively thinner. Yeah, that's right. And D. The movements of each eye are controlled by 6 muscles, not 4. There are 4 rectus muscles and 2 oblique muscles, making a sum of 6. Getting to the photoreceptors. The photoreceptors are located in the retina at the back of your eye. There are 2 major types of photoreceptors, rod cells and cone cells. Rod cells have an absorption peak in blue-green light. That is to say, the blue or green color of light can activate the rod cells to a greatest extent. Therefore, we sense a Purkinje shift. That is to say, when we use rod cells only and not cone cells, we will sense that the colors that we see are somewhat bluish or greenish. This is especially true when we are in dim locations, because rod cells are much more sensitive to light. It is responsible for scotopic vision, which means vision at night or in dark locations. That's why the Purkinje shift usually occurs at night. On the other hand, cone cells, there are 3 types of cone cells: L cones, which are long wave sensitive, M cones, which are medium wave sensitive, and S cones, which are short wave sensitive. L cones are responsible for sensing red light, M cones for green light, and S cones for blue light. Therefore, cone cells are the cells that help us sense color. Cone cells are also less sensitive to the brightness of light. We need a stronger light to activate them, so this is photopic vision, usually functioning in the day. And finally, there is another type of cell that are not strictly photoreceptors, but are also sensitive to light. There is another type of cell that are strictly not photoreceptors, but are also sensitive to light. They are the intrinsically photosensitive retinal ganglion cells. They are responsible for the light entrainment of circadian rhythm. In other words, they sense whether it is light or dark and tell our brain whether it's day or night. This is the structure of a rod cell and a cone cell. It has 3 major parts: the outer segments, which have many discs in them, the inner segments, which contains the nucleus, and the synaptic terminals. Phototransduction. When in the dark, the photoreceptors are actually depolarized. This is somewhat surprising. In the dark, guanylyl cyclase is active, which increases the concentration of cyclical GMP. These cyclical nucleotides open sodium channels, which allows sodium and calcium to flow in, and depolarizes the cell. This keeps the membrane potential of photoreceptors at around -35 microvolts, which is higher than the normal resting membrane potential. When there's light, something happens. There are some proteins called photopigments inside the photoreceptors. They are opsin proteins that are bound to chromophores. Chromophores are chemical molecules that have color, which means that they can absorb some wavelength of light. In human, the chromophore is always 11-cis retinol. The 11-cis retinol, which is stuck inside the opsin, isomerizes when it receives light. The process is called photoisomerization. It turns into this all-trans retinol, which is not crooked but straight. The conformational change of this molecule causes a conformational change of the whole opsin protein and activates the opsin. The opsin is a G protein coupled receptor and activates downstream reactions. When the opsin has gone through this process, it gradually recovers from trans to cis via enzymes. This process is called bleaching. iPRGCs also use opsins, but they use a different variety of opsin called melanopsin. Every opsin has a peak in the spectral sensitivity, meaning that it is most sensitive to a specific wavelength of light. L, M, and S cone cells have opsins that have different sensitivity: red, green, and blue. And as you can see in the dashed line here, the rhodopsin, which is used in rod cells, is somewhere between green and blue, which we talked about it before. This causes the Purkinje shift. Now, when opsin is activated, the light causes the photopigment to activate, which causes the opsin, the G protein, to activate, and the G protein, in turn, activates the phosphodiesterase, which decomposes cyclical GMP. This causes the inflow of sodium to diminish and hyperpolarizes the cell. In short, darkness depolarizes the photoreceptor and light hyperpolarizes the receptor. Photoreceptors also play a part in adapting to light and dark. In the dark, the adaptation is mainly through the regeneration of unbleached rhodopsin proteins. In the light, we can see this. Usually, calcium, which inflows through the sodium channels, inhibits guanylyl cyclase. This keeps the depolarization under check in the dark. However, when there is light, there is less inflow of calcium ions. This releases guanylyl cyclase from inhibition gradually and helps us adapt to bright environments. This is a summary of what we've reached today. We have seen the process of light transduction in photoreceptors. That is the first step in vision perception. A final question time. Please select the correct statements. A. The Purkinje shift can be explained by the spectral sensitivity of rhodopsin in rod cells. B. Light transduction uses cyclic nucleotides as secondary messengers. C. Cone cells can sense light because every cone cell contains more than one type of opsins. D. iPRGCs participate in non-image forming functions. The correct answers are A, B, and D. Why is C wrong? Well, cone cells can sense color not because every cone cell contains more than one type of opsin, but rather because there are several types of cone cell. Each type of cone cell contains one specific type of opsins, but these opsins differ between types of cone cells. There are cone cells for red, green, and blue color sensation. In this episode, we have had an overview of sense and perception, an overview of vision, and take a look at the eye and the photoreceptors. We will dig deeper into the retina from the photoreceptors in the next part of this episode. See you again in part 9-2, Vision Part 2.